Now we start with the fifth hormone, plant hormone, that is abscisic acid, which is also known as ABA. It is actually a plant growth regulator, but it is actually a inhibitor. It is a growth inhibitor. Discovered almost simultaneously by Endicott in 1963 and Waring in 1964, but they named it abscin 2. They named it abscin 2 and Dormin respectively. So abscin 2 named by Endicott and Dormin named by a Waring. So later on it was decided to call as abscisic acid. Okay, so this is about the some of the particular point of the auxin. If we talk about occurrence, then it is found in all group of plants. Found in all group of plants, from mosses to the higher plant. From mosses to higher plants. Okay, so it is about the occurrence. One more important point that you should remember, in liverworts, an algae compound linoleic acid has been found having activity similar to abscisic acid. Having activities similar to abscisic acid. When we talk about abscisic acid, it is a major growth inhibitor in plant and antagonistic. It is antagonistic to all the three growth promoters. And especially gibberellic acid. It is especially antagonistic to the gibberellic acid. When we talk about precursor of this abscisic acid, then that is viola xanthin, and viola xanthin is nothing but a type of xanthophyll in chloroplast, produced mainly in mature leaves. Besides this, it is also synthesized in stem, fruit, seeds, and then transported to the rest of the plant through vascular tissue. So this is the thing which is found in case with respect to the abscisic acid. Now next we talk about the functions of abscisic acid. So next is functions of abscisic acid. Now when we talk about function, as its name suggests, first function is abscission. Abscission means it promotes falling of leaves, flower and fruit. So it hastens this process, promote abscission. Second, it helps in closing of stomata. So it help in closing of stomata by potassium ion leave the guard cells. So when this potassium ion leave the guard cells become flaccid and they lost their rigidity due to the shortage of water and rot. Hence, it is known as stress hormone. Abscisic acid is also known as stress hormone. Third, it promote bud dormancy during winter. It promote bud dormancy during winters. And fourth, that is, it induce seed dormancy. Hence named dorming and help in seed. So it help seed to withstand desiccation and other unfavorable condition. Okay, after this dormancy, fifth is stoppage of cambial activity. So it inhibits the cambium activity of vascular cambium. Sixth, if we focus on this abscisic acid induced flowering in some short day plant like strawberry and blackcurrants. Seventh is play important role in abscisic acid play important role in seed development, maturation and dormancy. Okay, that is also one of the important point with respect to abscisic acid. Next point is it induce synthesis of carotenoids in green oranges, making them yellow. So it become yellow due to this. So these are the particular functions of the abscisic acid. And last we talk about bioassay, that is cotton or bean explant test. Bioassay of abscisic acid is cotton or bean explant test. So now after studying all the plant growth regulators, now we are moving to the last some of the topics like dormancy, then flowering, photoperiodism and vernalization. So now first we start with the dormancy. So when we study dormancy, It is defined as it is a inability of viable seeds to germinate due to internal factor under favorable condition under favorable condition, then it is known as dormancy. So dormancy, it is a suspended growth due to the internal factor or inability of viable seeds to germinate due to the problem in an internal factor or intrinsic factor. Now after dormancy, one more term we discuss here, that is quiescence. So it is inability to germinate or suspended growth due to external factor, means under unfavorable condition, then it is known as quiescence. So quiescence is due to the defect in the external factor and dormancy is the defect in the internal factor, then these two terms are observed. So dormancy occurs, dormancy is a one type of suspended growth. So next we discuss what are the regions of dormancy. When we study regions of dormancy, we come to know that it can be due to seed coat. It can be due to seed coat. So seed coat can be impermeable for gases. And when seed coat is impermeable for gases, then also dormancy can occur in case of apple. So apple is the example. Second, seed coat impermeable to water, then seed dormancy observed in case of trigonella because water will not enter. Next, seed coat is mechanically resistant, and if seed coat is mechanically resistant, example are capsula and amaranthus. So this way also, due to seed coat also, dormancy can occur in case of these different plants. Another region can be due to presence of immature embryo. Example is ginkgo biloba. In ginkgo biloba also, the dormancy occurred due to the immature embryo. The dormancy can also occur due to specific light requirement. And in specific light requirement, based on this light requirement, the seeds are of two type, that is positively photoblastic seeds positively photoblastic seeds and negatively photoblastic seeds. When we talk about positively photoblastic seeds, these seeds require light for germination. This required light for germination. They are positively photoblastic cells. An example are Lactuca sativa, then Nicotiana tabacum, and Lythrum. Okay, in all these are positively photoblastic seeds. But when we talk about negatively photoblastic seeds, this seeds show inhibition of germination on light exposure, and the example are onion, lily, and phlox. Okay, so these are the particular positively and negatively photoblastic seeds. So due to specific light requirement also seed germination can be stopped. Next is demand of after ripening period. Demand of after ripening period. Some seeds have a mature embryo but do not germinate. Some seeds have mature embryo but do not germinate due to absence of growth hormone. Due to absence of growth hormone. So they require a period of after ripening during which they attain a power to germinate. So during after ripening period, the requirement of growth hormone will be completed and they will start to germinate. Example are oat, barley, and wheat. So that is the demand of after ripening period. Dormancy can also be observed due to presence of germination inhibitors within seed. And if we talk about germination inhibitors, the example are some chemicals like organic acids, phenolics, tannins, alkaloids, lactones, mustard oil. This all are the growth inhibitor. So they inhibit germination. Specifically, ferulic acid is an inhibitor present in tomato pulp. Okay, so these are the different regions responsible for the seed dormancy. So now we start with the methods to break dormancy. Now these methods include, first is scarification. Now scarification involves a softening and weakening of seed coat by acids, alcohol, or knife. Okay, whereas second method is stratification. Now in first method, if there is a softening and weakening of seed coat, then seed coat become permeable to gases and water, and seed germination can occur. Stratification involves after ripening treatment of low temperature, that is 0 to 10 degree Celsius with O2. Third, either you provide a light exposure for the positively photoblastic seeds, and fourth is you can provide a low temperature plus gibberellin, because gibberellin also stimulate seed germination and O2 treatment. So by these different methods you can carry out a breakdown of seed dormancy and seed germination can occur. So now next we are going to discuss seed germination. So next we talk about seed germination. So here suspended growth is over. Now when we talk about seed germination, first we should draw the diagram of a seed. Now we know that when there is a seed coat, it have a embryonal axis and cotyledon. These are cotyledon, this is plumule, this is epicotyle, this is hypocotyle, and this is radical. The region of embryonal axis between the cotyledon attachment site and the radical is hypocotyle, and cotyledon attachment site and plumule is epicotyle. So what happens first? Here radical grows first, and this radical growing after this, it established as a primary root, then later will be work as a taproot. So established within soil occur properly. After this growth of radical, hypocotyle or epicotyle growth. Then out of any two, any one will grow, then later on another will go, and the plumule growth will be observed. And after a plumule growth, seed coat get released and it will be established as a plant sapling. So this way particularly the seed germination can occur. So now we need to talk about the types of seed germination. So when we talk about type of seed germination, there are of two types: apigeal and hypogeal. Now in apigeal, hypocotyle grows first, so cotyledons come out of soil. Suppose this is the layer of soil and this is the seed coat, and this seed coat have a embryo. Now this primary root will develop from this. After this, if apigeal germination will occur, hypocotyle will grow first. Hypocotyle is here this, so this if hypocotyle grows, then cotyledon will come out of the soil. Okay, but in hypogeal germination, epicotyle grows first, and when epicotyle grows first, cotyledon remains within soil. So here if you see, this is the surface of soil, this is the cotyledon, then what here will happen, this if epicotyle grows, then epicotyle will grow and reach to the out of soil. It will develop a leaf, okay, but cotyledon will remain within soil. Why? Because epicotyle is growing fast. This epigeal germination is seen in case of cucurbits, mustard, castor, onion, tamarind, etc. Whereas in case of hypogeal germination, it is found in case of rice, maize, mango, fabaceae, etc. So these are the different type of epicotyle and hypocotyle germination. Now after this we discuss one specific type of seed germination, that is known as vipary or viviparous germination. Now in this viviparous germination, seed germinate inside fruit. So this occurs when seed germinate inside fruit, when fruit is still attached to plant, and this type of mechanism is found in case of mangroves plant like Rhizophora, Soneraceae and Heritia. So now the question is, why it occur in mangrove plants? Because mangrove plants are found in saline areas, and in saline areas what we find, the salt concentration is. So what happens when fruit is there and within seed is there, if fruit reach to the soil and seed become available, the outside saline condition is there, so water will be pushed out, and due to which the seed will not germinate due to unavailability of water or deficiency of the water. So if this will keep continuing, this plant will extinct because the seed germination and propagation will not occur. So what they have evolved, they started germinating this seed when present within particularly fruit over the plant. When their weight will increase, they will fall on the soil, and within soil they will establish and will grow as a new plant. So this occurs in case of marine or saline habitat, salty habitat of a mangrove plant. Okay, so this is with respect to seed germination. Now next topic which we are going to discuss, that is flowering. Now flowering involves production of flower, and if flowers are formed, means it is a part of reproductive phase. Now in this flowering, what happens? Shoot apex or apical bud convert into floral bud, and this floral bud will form a flower which can occur by a regulation harmonically. Now flowering is influenced by two things, light and low temperature. So flowering phenomena we are going to discuss, that is photoperiodism and vernalization. These are the two phenomena which are responsible for the flowering. First we are going to talk about the photoperiodism. Photoperiodism is the requirement or need of specific light period for flowering. Now in this light period is also known as photoperiod or scotoperiod. So it is a specific time requirement. It is the availability of hours of light for this flowering. It is known as photoperiodism. Now when we talk about this photoperiodism, the response of a plant in this response of plant to changes in relative lengths of day and night is also called as photoperiodism. Now, so the relative length of the dark and light period is vary from place to place and from season to season. And this length period, as we know, it is known as photoperiod. At equator, day length is of 12 hours duration throughout the year. Plants flower only when exposed to more or less than a certain light period, called a critical photoperiod. So next we talk about a critical photoperiod. Critical photoperiod is a photoperiod or a period in which plant flower only when exposed to more or less than a certain period. More or less than a certain period, called critical photoperiod. Now this critical photoperiod also can vary for the different plant in different area. So according to the photoperiodic requirement of plant, next we are going to discuss types of plants. And when we discuss the different types of plant, first we talk about short day plants. Short day plants can also be called as long night plants. Okay. So short day plants required light period less than critical period. So they will flower only when the light period is available less than the critical period. Suppose critical period is of 9 hours then of that particular area, then all the short day plant will flower if they get a light of either 1 hour, 2 hour, 3 hour, 4 hour, 5 hour up to 9 hour. If they get a light beyond 9 hours, they will not be able to flower. So this is the critical period, that up to which they can flower and below which they can flower, but not beyond that. Okay. So if we take an example of this short day plant, then we write xanthium, which is known as cockleber, rice, chrysanthemum, sugarcane, coffee, soybean, and cosmos. These all are the different short day plant which cannot cross and cannot survive a flower if the light beyond critical period is provided. Second type is long day plants. Now long day plants are also named as short night plants. So long day plant will flower when they exceed the critical photoperiod. Means when they get a light availability more than a photoperiod, more than critical period, then only they can flower. An example of this are henbane, that is Hyoscyamus niger, spinach, radish, sugar beet, oat, barley, wheat, and larkspur. These all are the long day plant or short night plant. Okay. Third is day neutral plant. Day neutral plant are DNP. Their flowering is not dependent on photoperiod or light period. Example include cucumber, tomato, maize, cotton, sunflower. There are some other type of plant like SLDP. They can be called as a short long day plant. Now short long day plant means they initiate flowering in short day period, but flowering or blossoming actually occur in long period. Same way, there is another category of long short day plant. In long short day plant, they initiate flowering in long day period, but flowering or blossoming occur in short day period. So these are the different categories which are also found. But the stimulus of light period is received by pigment called phytochrome, that is carried out by the pigment called a phytochrome. Now phytochrome, it is a blue-green pigment, exists in to interconvertible forms. That are PR and PFR. Now PR is also known as P660 and PFR is also known as P730. Means PR converted into PFR by absorbing red light, because PR absorb red light and PFR absorb far red light. So this PR converted into PFR by absorbing red light, but on absorbing far red light, the PFR converted into PR. In dark period also, PFR get converted into PR, and out of which the PFR is physiologically active form and PR is particularly physiologically inactive form. PR is found in cytosol and PFR is attached to plasma membrane. Okay, so this is the different forms which are observed in PR and PFR. So when we talk about this PFR and PR, when dark period is there, the PR is going to accumulate, and when light period is there, PFR is going to accumulate. So when we calculate this ratio for the short day plant and long day plant, we come to know that for short day plant, in short day plant, short day period or day period is short, means the dark period is light, because they are also known as long night plant. And if dark period is more, then PR get accumulate. So PR upon PFR ratio become greater than 1. And for long day plant or short night plant, this PFR get accumulate. So PR upon PFR ratio become greater than 1. And these are the ratio which are critical for flowering. So that you should remember. Now, photoperiodic stimulus is received by leaf, and when proper photoperiod is received, a florism hormone is synthesized. In proper photoperiod, florism hormone gets synthesized in leaf. Then this florism are transported to bud through phloem and flowering occur. So this is the thing which occur during the photoperiodic stimulus. Now it is considered that florism is a hypothetical hormone, and this is chemically similar to gibberellin. Chemically similar to gibberellins. Chemically similar to the gibberellin. So that is the thing for the important role of pigment and hormone in this photoperiodic response. Now we should understand how the photoperiodic stimulus work and break reaction work for the short day and long day plant. Now when we consider a short day plant, it is also known as long night plant. Then for this, a normal stimulus, if we consider, then in 0 to 24 hour, if it is a short day plant, suppose 0 to 9 hour is a day period and this long night plant is 9 to 24. So this is will be observed in short day plant. Now in this short day plant, day period is short and dark period is long. Now actually night period is necessary for flowering in short day plant. So if this 0 to 9 period is there, but this period is broken up by the light stimulus or flash of light, or dark period is broken up by the flash of light, then flowering will not occur. But if this 0 to 9 period, 9 hours light period is there and it is a dark period, now this day period is broken by dark stimulus or dark period, then flowering occur. Because if dark period will increase, then it will obviously a light period will be shortened, and short light period is necessary for the short day plant, so flowering will occur. Now in short day plant, if we give different stimulant like 0 to 9 hours and 24 hours, and in this dark period also we give two break reactions. Okay, first we give a red light, then we give a far red light. Now in red light, what is going to happen, that PFR is going to accumulate. Okay, but when again after that we will give a far red light, in far red light what is going to accumulate, PR is going to accumulate. So if PR will accumulate, PR upon PFR ratio will be greater than 1, so flowering occurs. And in this case, in such case, in multiple responses, last stimulus is taken into consideration. After this, 0 to 9 and 24 hours given, and in this first we give far red light, then we give red light and we cause a break. In this case if we give a far red light, PR will accumulate, then we give a red light, so PR will accumulate into PFR. So now the ratio become PFR upon PR greater than 1. That will be important for the long day plant. But PFR upon PR ratio greater than 1 cannot be successful for flowering in short day plant, so no flowering. So such a way the particular different break reaction work in case of short day plant. But when we talk about a long day plant, in long day plant they are also known as short night plant. Long day period is necessary for flowering. So in this, if we see, if 0 to 13 hours is a day period and 13 to 24 hour is a dark period. Okay, sorry, 0 to 13 is a day period and 13 to 24 is a dark period. Suppose we are taking it 14 hours. So 14 hours of day period is there, long day period is there. So in this long day plant, long day period is necessary. So if we break this dark period, with light influence, no flowering, then flowering will not occur. Okay, because night period has broken up and day period has increased. Sorry, we have broken up dark period and light period we have given. So light period has increased means flowering will occur. But in this, if we break this with a dark period, flowering will not occur because they need a long day period, and here the day period becomes shorter. Okay, in this also, in dark period, if we give a red light, then far red light in case of dark period, and we break it, then initially red light will accumulate what, a particularly PFR, and then when we give a far red light it will accumulate PR. In long day plant, PR upon PFR ratio greater than 1 is not responsible for flowering, they need a more PFR, but in this case PR is going to accumulate, so no flowering. But if same condition is there and 0 to 14 hours period is there, and in 24 hours also we are doing a break reaction, and in break reaction the red light is far red light is given first and then red light is given, so far red light influence will be broken up by the red light, and in red light we know that PFR will accumulate, and for long day plant PFR upon PR ratio greater than 1 responsible for flowering, so here flowering will occur. So this is the photoperiodic responses, break reaction, and whatever happen to this particular photoperiodic way which is responsible for flowering. Next we are going to talk about next topic, that is vernalization. So after photoperiodism, next topic and last topic in this chapter which we are going to discuss, that is vernalization. This vernalization is also known as yerovization. This vernalization or yerovization means requirement or need of low temperature for flowering. This vernalization is the phenomena in which flowering is qualitatively or quantitatively dependent on exposure of low temperature. Now the stimulus is received by embryo or shoot apex, whereas in photoperiodism we have studied that stimulus is of photoperiod is received by the leaf and then transferred to the shoot apex. Here mature embryo or shoot apex receive the stimulus. Now there can be a annual plant and biennial plant. In annual plant they complete their lifespan in case of 1 year or 1 season. Now there can be two type of variety, that is winter variety and spring variety. Suppose now if we talk about the seasons of India, then there are 6 seasons like spring, then summer, then monsoon, then autumn, then pre-winter, winter and spring. So suppose there are two varieties, winter variety and spring variety. So when winter variety grown in winter or sown in winter, it will flower near to summer or monsoon and it will produce a flower happily. And if you talk about spring variety, it planted in the spring, it will flower in the summer and set the seeds and fruit and harvested during the monsoon or autumn. So this is often occur to the winter variety and spring variety normally. But suppose winter variety is normally sown in this spring season, but if it is sown in a winter, it will not flower. So if winter variety is shown in spring, it will not flower because its requirement of low temperature has not completed. So if we artificially provide low temperature and then sown in spring, they will flower. Means they actually need a low temperature. And if we do the second thing, that artificially we provide a low temperature and then we sow in the spring and it will flower, then this time period get reduced in the second case. So it will also help to reduce the time period of flowering. So it means artificial cold treatment, the biennial and many winter varieties of cereals can be made to flower in same season. So this is the vernalization through which the time period of flowering is reduced. Term is given by Lysenko. Site of vernalization are shoot tip, embryo tip or root apex that we have discussed. Now we discuss the requirement of low temperature or requirement of vernalization. When we discuss the requirement of vernalization, we come to know that it required low temperature, that is 0 to 5 degrees Celsius. Second, period of low temperature, that is few hours to few days. Then third, we required actively dividing cells. Fourth is required water. Fifth is required aerobic condition. Sixth requirement is proper nourishment. So these are the requirement, and in this process low temperature treatment is a hypothetical hormone is produced, and in vernalization, in this hypothetical hormone is produced known as vernalin, okay, during the low temperature. And this is now believed that Vernalin is a gibberellin like substance because vernalized plant have higher gibberellin level. Okay, now vernalization is beneficial in reducing period between germination and flowering. So due to which thus more than one crop can be obtained during a year. So this is the thing of vernalization or a low temperature requirement. So now all the topics with related to this chapter are over, and I hope you have understood. Thank you.